The big challenge here is this natural history with this dramatic degree of interpatient variation. We've talked about the waxing and waning of steatohepatitis here, which is presumably subject to a degree of modifiers, including genetic modifiers and also environmental modifiers. And then presumably there are further modifiers which will come in here, which will determine not only the rate of fibrosis progression, but indeed whether fibrosis progression will occur or not. And so one of the key questions we have to answer first of all before we consider the role of genetics as a potential biomarker is, are there actually heritable factors that we could use to risk stratify patients into these prognostic high or low risk groups? And the answer is a resounding yes. We've got three very strong lines of evidence to support the role for heritable control of inter-individual variation: familial aggregation, inter-ethnic differences in susceptibility, and a number of twin studies comparing monozygotic and dizygotic twins. I'm just going to show you this very nice data from Rohit Lumba's group in UCSD, where he compared as I say, monozygotic and dizygotic twins, and demonstrated a really surprisingly high level of heritability, not just for steatosis, but also for fibrosis, with approximately 50% of the variation for both of these phenotypes coming from heritable factors. And so the question then is, well, where might that heritability stem from? And I think we all remember from our undergraduate days the idea that DNA makes RNA makes protein. And I'm going to focus in on a number of these. I'm going to particularly deal with DNA sequence variation and DNA methylation. And I know that Professor Frank, who will be speaking next, is going to be covering microRNAs. So let's think first of all about what we mean by a biomarker. And this is something that's, again, been highlighted during the day. I just want to plant this in our minds now before I review some of the genetic data, so that as we're going through this, we can be thinking about, well, how can the genes fulfill these specific criteria? We're looking for diagnostic markers, so degree of steatosis, severity of fibrosis, those sort of things. We're looking for prognostic markers to stratify those individuals into fast or slow progressors, high or low risk, and to predict long-term outcomes and hard endpoints such as hepatocellular carcinoma. We're looking for predictive biomarkers. Now, this is an interesting one. Can we spot particular groups of patients who are more likely to respond to one therapy over another? And that's a very interesting area. And then we're looking for these monitoring dynamic biomarkers that allow us to track disease severity or treatment response. And I think it's probably no surprise that in terms of a SNP variation, it's very unlikely that we're going to see a dynamic marker in terms of SNP variation. So we'll put that one to one side for a moment, but we'll come back to it again later on in the talk. So what do we know about genetic variation so far? Well, for a long time, the majority of our understanding has been based on candidate gene studies. And so it's not surprising, given the nature of candidate gene studies, that you set up a hypothesis. I think insulin resistance is important. You look for genes that may influence insulin resistance. And sometimes you find them, and sometimes you don't. And as you can see there, based on our understanding of pathophysiology, we've got genes for insulin sensitivity, fatty acid flux, oxidative stress, endotoxin response, cytokines, and fibrogenesis. But if we filter that list down to those genes that have been independently validated in multiple cohorts, or where multiple different statistical techniques have been used, the list becomes substantially shorter, and it's just the ones in bold there. So IRS1, EMPP1, FADS1, GCLC, SOD2, and KLF6, a much shorter list. And I want to highlight apolipoprotein C3, which very high impact publication at the start, got a lot of attention. Since then, 4 separate studies, 4,000 individuals across multiple ethnicities, unable to validate that association. And so we have to put ApoC3, unfortunately, into the false positive association box. The real advantages and the real advances here came with the genome-wide association study. What are the problems with it? It requires a large sample size. Now, that's fine when we're looking at blood pressure measurements or if we want to quantify steatosis. But if we want to look at histological steatohepatitis or fibrosis, it's a lot harder to build up a big cohort. And it's taken a bit of time to do that. We all know the process: big cohort, multiple SNPs tested, lots of chi-squared tests with a conservative p-value, but the need for validation. And it's important to remember that genome-wide association studies find loci, they don't find a given gene. It's up to us to then editorialize and decide which gene in that region is the causative gene. There have been a number of studies in fatty liver disease. The largest of these have all used steatosis, quantified either by CT or MRI, as the main phenotype. And that's understandable because it's a much more readily accessible phenotype to use. But it is, as I've mentioned, phenotype limited in terms of looking at other aspects of the disease. What really got the field going was, of course, this lovely study from Helen Hobbs and Jonathan Cohen, looking at the Dallas Heart Study cohort, with that single strong signal there on chromosome 22, which was the Pmp La3 RS738409 variant. And they demonstrated very nicely this stepwise increase in fat content with each copy of the allele. Subsequently, in histologically characterized cohorts, that same association has been demonstrated for steatohepatitis and for more severe fibrosis, here looking at a mixed UK and Italian population. And just to bring us right up to date, this is the phase 1 analysis from the FLIP-EPOS GWAS study, and again you can see that same very strong signal across all 3 histological characteristics for the first time at genome-wide significance levels. If Pmp La3 is associated with progressive cirrhosis, it's then also tempting to wonder, well, could it also be associated with other hard endpoints, such as hepatocellular carcinoma? And this is something we were able to look at in the EPOS cohort, where we have a large number of histologically characterized fatty liver individuals and also a large number of well-characterized HCC patients. And what we found was that carriage of the Pmp La3 variant was an independent risk factor for developing primary liver cancer, independent of age, gender, body mass index, diabetes, and crucially, independent of fibrosis stage. Carriage of 2 copies of the minor allele confers approximately a 5-fold increased risk of primary liver cancer compared to a background fatty liver population. And when you compare it to a population background, that becomes a 12-fold increased risk of developing cancer in individuals who carry 2 copies of this variant, which is striking to say the least. This same association has also been reported by a number of other groups, but unfortunately not able to control for background fibrosis in the majority of the other studies. But the message we're getting strongly is Pmp La3 is associated with cancer risk in steatotic liver disease, that's NAFLD and alcohol, but less so in non-steatotic liver diseases. One of the issues with genome-wide studies is that you find a gene, but you don't necessarily know what it does. And this has been an issue that's dogged Pmp La3 from the start. We know it doesn't make you diabetic. We know it doesn't make you obese. There was some data to suggest it may increase lipogenesis, but then there was some data to suggest that maybe you reduced triglyceride hydrolysis. And this has continued really throughout, looking at animal models and so on, and there's still significant debate about what it actually does. But we are beginning to piece this together. And Helen Hobbs, again, has helped us here. Very nice study overexpressing the I148M variant in hepatocytes. And I stress that, in hepatocytes in mice. And you can see here in greater fat content in carriers of that variant. And when she looked at gene expression, again, we saw a mixture of differences in gene expression associated with fatty acid handling. And so the concept that Pmp La3 may play a role in remodeling of triglycerides came to the fore. But it's also telling what wasn't seen. There was no overexpression of procollagen 1, alpha smooth muscle actin, or TNF-alpha, so markers of steatohepatitis or fibrosis. And so that bridges the question, do we need something else here, a second hit? Or does it matter what cell type you're looking in? And Helen, this paper was only looking at hepatocytes, as I mentioned. So at that point, the field stalled for a little while. There was some very nice data that came out from Stefano Romeo indicating that the 2 tissues where Pmp La3 is most highly expressed is the retina and the liver, connection, possibly retinol metabolism. And then this paper came out just a couple of weeks ago in press, and this finally begins to pull this all together. And it also speaks to one of the comments we got from the floor earlier on today with a question about why are these metabolic genes coming through to fibrosis. And so this is data from Michael Trauner's group in Austria, where the first thing he's demonstrated is that Pmp La3 expression increases in primary hepatic stellate cells alongside stellate cell activation. So the two go up in parallel. What was also telling here is that if you silence Pmp La3, you get less stellate cell activation. So we then get the concept that Pmp La3 is necessary for stellate cell activation. And on top of that, carriage of the variant form confers a pro-inflammatory and pro-fibrotic profile. And so this picture builds up, and we get the concept that the Pmp La3 variant is in fact acting differently in different cell types. Its expression in the hepatocyte drives lipid accumulation and potentially lipotoxicity. Its expression in this stellate cell, however, affects junk signaling. It causes disinhibition of stellate cell activation by downregulating PPAR gamma, and it also leads to the release of a range of cytokines as well as fibrogenesis. Finally, we're beginning to put this puzzle together. Very exciting and very relevant. If we move on through our quick trot through the genome-wide history, the next one came from Liz Spiliotti's group, Pmp La3 again, range of other variants such as GCKR, and this signal on chromosome 19, which was first called the NCAN gene. And this is an important one because this shows you how we can get it wrong with genetic variation. There's NCAN here, but each dot up here is a SNP, and the higher up they are, the more likely they are to be co-inherited. And what you've got here is a cloud of about 450 kilobases of variants that are all highly co-inherited in strong linkage disequilibrium. At one end of it is NCAN. At the other end of it are a number of genes that have been previously associated with dyslipidemia. And just here in the middle is TM6SF2. But the truth is, with a simple association study, it could be a variant anywhere in that bracket, and it's up to us to work out which one it is. It turned out that it wasn't NCAN, and this very nice study from Helen and Jonathan again confirmed this. It was a reanalysis of the Dallas Heart Study cohort, this time using an exome chip, and it pulled out the TM6SF2 gene on chromosome 19. Using a conditional analysis, they were also able to demonstrate it was definitely this particular variant, not the NCAN variant, that was most important here. But again, just looking at steatosis. We were very keen to see if this same gene would also associate with inflammation and fibrosis, and so looked in the EPOS cohort, and I'll just skip through that slide in the interests of time. But what we found was, once again, carriage of the TM6SF2 variant was an independent risk factor for advanced fibrosis, independent of age, gender, BMI, diabetes, and independent of Pmp La3. It's a less common variant, but it appears to be a more potent variant in terms of conferring risk for advanced liver disease. So, since then, a number of studies have confirmed our findings, but interestingly, there have been a number of other studies. And so the picture forms that if you carry the minor allele, you keep the fat in the liver and you're at risk of fatty liver disease. But there were also a number of studies showing that carriage of the major allele, the common allele, is associated with increased cardiovascular risk. Here's just one of those studies, and you can see the reverse association for carotid plaques compared to fibrosis or NASH, and this very nice demonstration of increased risk of cardiovascular disease if you carry the more common allele. And this was also validated in another GWAS by the Holman group. So, working with Liz Spiliotti's, we've put out this idea of the catch-22 hypothesis here. However you cut it, being overweight and diabetic is not good for you. It increases the flow of fat through the liver. But then TM6SF2 acts as a master regulator, determining how you respond to this metabolic stress. If you carry the minor allele, as I say, it's fatty liver disease. If you carry the major allele, you push the fat out, you spare the liver, but at the expense of a heart attack or stroke. Now, it's difficult to think how we're going to drug that successfully, because really it's a pick what you want to die from scenario. But nevertheless, as I think it would be reasonable to say, this may be useful because it dissociates cardiovascular disease and liver-related disease in fatty liver, and therefore maybe it indicates whether our patients need to see us in this room or see the cardiologist down the corridor. And this is a concept we need to think about. What does TM6 do? We're not really sure. It's overexpressed in the ER-Golgi intermediate compounds. Overexpression of it appears to influence hepatic triglyceride content. And there's been a large degree of data coming through in the last few months showing that it's preferentially expressed in the liver and small intestine. As I've mentioned, it can affect dyslipidemia. And importantly, it appears to affect the packaging of lipid and its excretion in VLDL droplets. But quite where it fits into that pathway, we're still really trying to drill down. The last gene I want to talk about very quickly is MBOT7. Unlike all the others, which have been identified in fatty liver and subsequently validated in alcohol, this one was identified in alcoholic liver disease and subsequently validated in non-alcoholic fatty liver disease. And here's the data from the Dallas Heart Study and also in an Italian-Finnish cohort demonstrating the carriage of the MBOT7 variant is associated with greater NASH, greater fibrosis, and also alterations in lipid handling, but within the hepatocytes themselves. So, to come back to the start of this then, can we use this to help us target the patient? Well, the problem here is, and we have to bear this in mind, this is a complex disease trait. This is not a monogenetic condition. This is not cystic fibrosis. And therefore, we are dealing with a condition where there are multiple modifiers, each with a relatively modest effect. I also want to highlight that all the data that exists at present are association studies and do not prove causality, and no data has been presented thus far that links any genetic variant with long-term outcomes or rates of progression. And that's something we're generating some data on within EPOS at the moment, but it's a key question. So, clinical application of Pmp La3. Well, there have been a few studies to diagnose fatty liver, to look for NASH, to look for the presence of fibrosis. And the bottom line of all of these is that adding in Pmp La3 on top of risk factors that we're already aware of doesn't add a great deal to the area under the receiver-operated curve, mainly 2 or 3% at most. And there's a reason for that, and this is it. These are complex genetic traits. The odds ratios for each of these genes, as I've shown you, it's in the order of 2 or 3. And so the false positive fractions with an odds ratio like that are in excess of 75%. And this is the challenge we're faced with. So it's unlikely that one genetic variant on its own is going to tell us the future or answer this question. And that's fine. It doesn't mean that there's no value to these, however. What we're seeing is that as we combine genetic variants, we're able to explore more and more of the heritability, here bringing together Pmp La3, TM6, and MBOT7. And we also need to consider whether we can use it for the negative predictive value. I showed you the data regarding hepatocellular cancer risk, and it's worth remembering that the rates of HCC in NAFLD are increasing exponentially. This is data from my colleague Helen Reeves in the northeast of the UK, and the orange bar here is the year-on-year increase in fatty liver disease in the north of the UK, which is a low viral hepatitis population, but we are seeing similar things in many territories. The positive predictive value, in fact, of carriage of the Pmp La3 variant for liver cancer is pretty low, but the negative predictive value becomes much higher. And so this will require a lot more validation before it's ready for use in practice, but if validated, it may help us in a situation not identifying the people who are at high risk of developing HCC, but identifying the people who are at extremely low risk, so we can save resources and don't need to put them into surveillance programs. More validation needed, more work needed, but an interesting concept. And then, what about informing precision medicine? Well, this is a really early area of the field, but we are beginning to get some indications that there may be genetic variants here that can influence long-term outcomes. We've seen some data suggesting a less beneficial effect with omega-3 fatty acids. We've seen less beneficial effects with statin therapy. On the other hand, we've seen a greater response to lifestyle change in individuals who carry the Pmp La3 variant. And And the final one there in the elafibranor study, and this was non-significant, and the power to answer this particular question in the Golden 505 study wasn't there, but we did see a non-significant trend at p equals 0.071 towards an enhanced effect in carriers of this particular variant. Early days, I think we're all looking forward to the big cohort studies coming through in the phase 3s, but an interesting signal nevertheless. So what about my question? Well, where can the genes help us? Insights into pathophysiology? Undoubtedly. Potentially novel therapeutic targets. Can they help us with diagnostics? Yes, it can. Probably not for presence of fatty liver disease. We can work that one out other ways. Could it help us identify people at greater risk of disease severity? Yes, but with some caveats. What about prognostic? Well, the idea of hepatotropic versus cardiotropic metabolic syndrome. We're getting that signal with TM6SF2. It helps us stratify towards risk of hepatocellular carcinoma and late complications. We're not there yet with the fast versus slow fibrosis progressors, but I think that data will be coming along. And then predictive. We're beginning to get those signals that genetic variants may influence how we respond to treatment. I left monitoring and I said I didn't think that coding variation was going to answer that one. In the last couple of minutes, I just want to very quickly talk about some epigenetic factors, and particularly DNA methylation, that I think gives us some interesting insights here. So we're all familiar with DNA methylation. An unmethylated gene promoter leads to transcription. A methylated gene promoter represses transcription of a given gene. So it's like Post-it notes. You can pop them on a gene, turn this one on, turn this one off. This is a very provocative study that my colleagues Jelena and Derek Mann published a couple of years ago in Nature Medicine. There was an argument, really. Evolution can't operate quickly enough to allow us to adapt to a new insult such as obesity. So there must be other ways that a population can adapt. So what did we do? We took some male rats. We treated half of them with carbon tetrachloride, the other half we didn't. We let them recover. We treated half their offspring with carbon tetrachloride, half of them we didn't. We let them recover, and we treated all the grandchildren with carbon tetrachloride. And what we saw was that individuals whose father and grandfather had been exposed to the same toxic insult causing liver fibrosis developed significantly less fibrosis than individuals who were the first generation to receive this particular toxic insult. And going on from that, when we looked at DNA methylation in the PPR gamma gene, which is a negative regulator of stellate cell activation, we saw changes in the methylation in particular CPG islands in the promoter region. Very exciting, probably worrying if you're a rat, but no more than that. However, we then went on and looked at a small cohort of individuals from our fatty liver clinic, comparing those with mild fibrosis and those with significant fibrosis, and we saw exactly the same methylation changes present in the same CPG islands in the PPR gamma promoter region. So that becomes quite challenging, and it sends out a difficult public health message in terms of the fact that if we all go to the bar after the lecture here, we're not doing it for us, we're doing it for our unborn grandchildren. But it's undoubtedly the case that we're seeing the same sorts of patterns in a number of other genes that are highly relevant to stellate cell activation and fibrosis. And I particularly draw your attention here to PGF-alpha, which I think we're going to be seeing a lot more interest in. And I think that's an important signal we're seeing down here. Great, but that still required a liver biopsy to do that data. We need to go beyond that. We need to find a way of detecting these signals in the circulation so we can have a blood test. And this is some data we've got in press in gut at the moment, where we've looked at circulating cell-free DNA. So this is extracellular DNA, which you can pick up in the blood, and we're once again seeing the same DNA methylation changes in the circulation that were detected in the liver. And in those same patients, we've got their biopsy tissue, we've gone in, we've used laser capture dissection, and we're seeing similar changes in methylation. So we're tying a peripheral blood signature of DNA methylation to a central change within the liver. Exciting. This requires a lot more validation again, but an interesting idea, and one which potentially will respond dynamically to increasing insults or indeed resolution of injury. Thank you. So to conclude, NAFLD, it's a complex disease trait. We need to avoid thinking of it as a monogenetic disease. I don't like the concept of Pmp La3 NAFLD. It's a very appealing idea in terms of it seems easy, but the truth is Pmp La3 is there in a population. Some people carry it, some people don't, and it's part of a constellation of risk factors, and we need to view it as such. We've got a number of coding variants that appear to be highly relevant here. There's some very exciting data on epigenetic modifiers coming through. I think we're going to hear a little bit about microRNAs in a minute, but DNA methylation as a peripheral blood signature I think is very interesting. There's still a huge amount of work to do to understand and identify more of these modifiers that undoubtedly are there, and then to translate these so that we can apply them as biomarkers. Just leaves me to acknowledge my colleagues, particularly mention the EPOS and FLIP consortia, who we've all collaborated very heavily together on this work, and to thank you for your attention.